The overall goal of this procedure is to compare the efficacy and toxicity of new RNAs targeting HIV-1 production.
This method can help identify new RNA molecules for use in HIV-1 gene or drug therapy, such as short hairpin or small interfering RNAs, ribozymes, and RNA decoys.
The main advantage of this technique is that several new RNA designs can be screened simultaneously with a quick turnaround time.
Although this method was designed to evaluate new RNA drug or gene therapy for HIV, it can also be applied to other systems such as small molecules or CRISPR-Cas.
We first had the idea for this method when we were setting up experiments to screen a large number of ribozymes for anti-HIV effects.
To begin, prepare a suspension of human embryonic kidney 293 T cells at 2 × 10⁵ cells per milliliter in DMEM with 10% FPS and 1% penicillin streptomycin. Add 500, 100, and 1000 microliters of the cell suspension to each of the wells of 24, 96, and 12 well plates for viral production, cell viability, and immune activation assays, respectively. Gently swirl the plates and incubate them overnight at 37 degrees Celsius with 5% carbon dioxide to 50 to 70% confluency. For the viral production assay, prepare a 10 nanogram per microliter dilution of an HIV-1 expression plasmid and add 10 microliters to each assay tube. Next, prepare 5 micromolar dilutions of test RNAs and a negative control RNA. Then, add 2.5 microliters or 10 microliters